All right, I'm going to start with the introduction. So it's my pleasure to introduce today's joint MindCORE and Computational Neuroscience Initiative Seminar Series speaker, uh, Bill Bialek. So Bill is a professor of physics at Princeton University, and he's also a member of the Louis Sigler Institute for Integrative Genomics. Um, Bill has really made fundamental discoveries in physics and biophysics and biology, and the area that he's been working on is actually very vast, ranging from questions in just general principles of why living organisms are organized and function the way they do, to more specific questions about spiking properties of neurons or cellular and molecular mechanisms and dynamics. So to me, actually, Bill has been a really important influence because when I just graduated from undergrad and was about to start graduate school, I was selected to attend a course at Woods Hole, Methods in Computational Neuroscience, and which Bill organized for many years. And that was really kind of a rite of passage for computational neuroscientists because the course brought together, and it's still going strong, but it brought together many people who keep coming back year after year. And many of us, actually, the people who we met at that course, we stayed in touch. And for example, Nicole Rust is one of my colleagues still here. We met at that course in Woods Hole, one of my collaborators at this hole. And Bill has really been bringing together people across disciplines and especially physics and biology for decades. He's also organized at physics a series of lectures that would bring together physicists and biologists. And he also organized a really important educational shift in how science is taught by building an integrative curriculum where physics, biology, and life sciences were taught together as kind of a core curriculum rather than with math, rather than as distinct a series of courses. Another aspect that's very influential is Bill's books that he's written. One of them, Spikes, probably many of you have picked up. And if you haven't, you should really read this as it's a fun read, but it also outlines really core principles of neuronal computation, as well as a book on biophysics and computation that came out more recently. So thank you so much for joining us.
Thank you. It's a pleasure to be here. It's been a while. There, I'm louder now. It's been a while since I've been at Penn, and it's nice to be back, see people I know, but also meet new people. I'm a little unsure about the audience. So I don't know what you know and what you don't know. And so I hope that we're not so many people that you feel inhibited from raising your hand and interrupting if something's not clear. So please do. It's better that I cover a little less material and be a little clearer than the other way around. So I've been interested for a very long time in a class of problems in, let's say, you could say a class of problems in biology, but where it seems that biological systems perform at a level which is close to the limits of what the laws of physics allow. And this is very different from the usual view. We often talk somewhat casually about biological systems being messy, being noisy, being irreproducible, and so on. But there are really important counterexamples to that. And so the question you should have in your mind is, are there enough of those counterexamples that you start to think, well, maybe that's the default, and when we see something that looks messy, it's because we're not understanding what's going on? Or is it just that there are these few exceptions off in the corners somewhere, and well, okay, anything that can happen does, and so you happen to find one that you liked, don't pay too much attention. And that's the tension in the background. So let me dive in. One of these presumably does something. Maybe. Put my glasses back on. Interesting. All right. So that works, sort of. Doesn't help very much. So here are two classic examples of near optimal performance, both from vision. So one, which I suspect many of you know, is that on a dark night, our visual system can count single photons. Now, the arguments for this go back to the early part of the 20th century, but the experiment that sort of got everybody to say, okay, yeah, probably right, is this famous one by Heckschler and Perrin in the early 1940s. There were other things going on in the world at the time, but somehow they managed to find time to focus on this. And essentially, the argument is that if you're looking at a very dim flash of light, sometimes you see it and sometimes you don't. And as I change the intensity, as I increase the intensity of the flash, it becomes more probable that you see it. And you might think that this is because of some internal noise source in the visual system, or because your attention is fluctuating or whatever. But there were reasons to think that maybe the origin of this variability is that when I control the intensity of a flash of light that's very dim, the number of photons that actually arrive at your retina will fluctuate from trial to trial, even if the settings of my optical source are the same. And so the idea is that you're willing to say, yes, I saw it, when you count up to some number of photons. And if that's true, then you'll see variability, because the actual number which you count fluctuates. A different example, actually, maybe it's best just to collapse. So if you plot the intensity of the flash in appropriate units, then if your model is that you're willing to raise your hand and say yes when you count 6 photons, then you would expect that your probability of seeing things follows the red curve, and the blue points are what people actually do, which is pretty good. Now, it's also true that they did this experiment in a regime where the area of your retina which is being illuminated contains hundreds of photoreceptors. And so if you're willing to say yes when you count 6 photons and they're spread over 100 receptors, the probability that any one of them got 2 is very small. So if this is the right interpretation of the behavioral data, you also know that individual photoreceptors must give reliable responses to individual photons. And it would take many years, but that's true, as many of you probably know. I would also point out that in order to be sure you absorb enough of the light, a single photoreceptor has 1 billion molecules of rhodopsin in it. So another way of thinking about what's happening here is that a photoreceptor cell can smell one molecule that has changed its structure out of 1 billion. So no matter how you slice it, this is a quite remarkable set of phenomena. And of course, it has an enormous set of consequences in the literature ever since. Let me go back to the other example. As soon as we understood something about diffraction in the late 1800s, and there was, for example, the theory of resolution in microscopes, somebody realized that insects look out at the world through much smaller lenses than we do. And so diffraction must be a much bigger problem. And that's true. So for example, an insect like a housefly has a resolution of about 1 degree, which is nearly 100 times worse than us, right? there's a trade-off in designing an eye, at least in designing a compound eye. Remember that in an insect, you have sort of the surface of the head, and you have lots of little lenses with approximately one photoreceptor behind each lens. If you make the lenses big, then diffraction isn't so much of a problem, but you can't fit many of them into the surface, so you just don't have very many pixels. If you make them really small, you get more pixels, but then eventually you get blurred due to diffraction. So the prediction is that if you look out at insects with different size heads, they should scale the size of the lenses to make the best trade-off between these things. And in the early 1950s, Horace Barlow went into the drawers of the Comparative Zoology Museum in Cambridge and checked. And he found that indeed the size of the lenses scales as the square root of the size of the head, and the slope is even approximately right. So this is telling you that the insect eyes that you see out in the world have been selected to essentially optimize their spatial resolution. There's another more cognitive example that I like, which has to do with recognizing whether something is random or correlated. So suppose I show you 10 coin flips, and I ask you whether that was a fair coin, or whether it was a weird coin that has correlations from one flip to the next. So if I only show you 10, and the correlation, there really are two possibilities. So correlations is not some gigantic category, it's some very specific form of serial correlation. And so I show you, and I ask you, was this random or correlated? Now, because there's only 10 flips, you can't do this perfectly. So there's a statistical limit to how well you can do, and that's what's shown in blue, and in red is how well people do. And you might think it matters whether the sequence looks symmetric or not, so they separated those out. It only makes a difference in one place. So mostly people get very close to the optimum. There's one thing that's really hard. So what's plotted here is the number of runs, that is to say, how many times do you see more than one thing in sequence? How many times do you see the same thing over again? And if you're constantly switching back and forth, so there's lots of runs, then because the thing which is correlated is positively correlated from one coin to the next, if you see alternation, the correct answer is random. But that's very hard to learn. When you look at something that goes heads, tails, heads, tails, heads, tails, heads, tails, you say, no, that's not random. Well, if the only alternative is positive correlation, then it is random. So that mistake you make. But for the rest, you do pretty well. So this is just, I mean, these are just a handful of examples meant by way of introduction. One can dig into each of them. They all have pluses and minuses. Even in the cases where there's strong evidence for optimal behavior, you could say, well, it's not quite optimal because of this, because of that. Anyhow, there's a large body of literature. But there's also an important oh yeah, so that was the little, I don't know if some of you remember the television commercials that advertised all this and more. It's also the name of a novel. There's an important counter thread, which is hard actually. The general feeling that biology can't be optimized is something that's very hard to put your finger on in literature. So where somebody comes out and says, no, 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 this can't be right, and here's why. So the closest is this famous paper by Gould and Lawton, and which I encourage you to read. It's very interesting, especially if you're interested in architecture. Essentially, it's an indictment not just of the notion of optimality, but of the whole notion of adaptation in the evolutionary sense. And their point is well taken, namely that we tend to look at things in the living world and say, oh, that's there because. And their version of this is, no, 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 you just don't understand how evolution works. It's not like that at all. I would point out there's another thing, which is that often when you read that's there because, there is actually no calculation that demonstrates that this is good for the thing that you claim it's good for. in which case, I don't know the content of the claim, right? So if you're saying, I don't believe you, I don't believe in your prejudice that things are there for reasons, well, that's a matter of opinion. But if you do a calculation and show that, for example, the size of the lens in the insect eye really is what it should be in order to optimize your spatial resolution, then that's true. Whether it generalizes or not, you don't know. So this is the argument that goes in the background of all of these things. And I would point out that, evolution is, of course, an interplay between randomness and selection. And so if you're on the side of saying that your default should be to think about what the optimal solution is, you're saying you think selection is more important than randomness. It's not clear that evolutionary dynamics would ever find the optimum in a realistic fitness landscape, and there are debates about that. Another way of saying it is, are the mechanisms that you see, clearly what you see today in organisms has traces of history, right? You can reconstruct evolutionary trees, but it's not clear, for instance, that the ability of, to give an example outside of neuroscience that I've thought about a lot, that the way in which information in a developing embryo is parsed out to tell cells where they are in relation to the main axes of the embryo, it's not clear whether the precision of that process is constrained by the history that the system took in order to get to the mechanism that we see today. That's a very different claim. In fact, as you may know, the best way to look to reconstruct evolutionary histories is to look at things that don't matter. Right? So if I look in regions of DNA sequence that, as far as we know, are neutral, then you can be sure you're reconstructing history and not variations in selection, right? So it's complicated. And one would also point out that when you talk about being constrained by your history, there's not only your evolutionary history, but the developmental history of individual organisms. Okay, so I hope this gives you some sense for what's going on in the background. What I want to do today is to revisit one example of the problem of optimization, which my colleagues and I thought about for a long time and have come back to recently for reasons that I hope will become clear. And then switch and talk about a more direct attack on this problem of whether it's possible to find optima or not. Okay, and I hope by the end you'll realize that a lot of intuition that many of us had about how hard it is to find optima was just wrong. And that has consequences. Whether they settle any of these issues or not, I don't know. So let's dig in. So the problem I'm going to talk about is the problem of how visual systems estimate motion. And it's a problem that sort of holds a special place in my heart because when I was a postdoc in the Netherlands, now before most of you were born, I had this wonderful good fortune to have the office next door to Rob de Ruiter, who was doing experiments and fly vision at the time. And it began a collaboration which continues. There is actually one after this. So I don't know. It might be that it proves that we're not smart enough to finish things, but it's also true that we've become wonderful friends. And it involved many people over the years. So here are some of the characters from the early part of our collaboration, and here are the more recent ones. And so let me, at the risk of telling many of you things you already know, let's talk about, if you wanted to estimate motion in the visual system, what computation should you do? Or what computation do you do? So naively, if something is moving, what that means literally is so that you see some spatial pattern of light intensity, which depends on the angle in the visual world and on time. And if it's moving rigidly, then that means that changing angle and changing time are the same thing. So if I take derivatives, they should be related to each other. And the problem of visual motion estimation is very easy. You take the derivatives and divide. So almost everybody in this room who does experiments knows that this can't be the right answer. And the reason is that when you're doing experiments and processing real data, there are two things that you try to avoid doing. One is differentiating, because usually there's more noise at high frequencies than at low frequencies, either in space or in time. And so if you differentiate, you tend to increase the noise relative to the signal. The other thing you shouldn't do is divide, because if signals are noisy, there's some probability you divide by 0. And you'll notice that this commits both of these sins all at once.
Very quick question.
Yeah. I think this is the wrong time to ask that question. It's not that it isn't a good question. It's just this isn't the right moment. Okay. So as many of you know, the alternative to this view goes back to experiments that were done by Reichart and Hachenstein in the 1950s with insects, beetles in particular, in a marvelous contraption where you let the beetle walk on this structure where periodically he comes to a Y and has to decide whether to turn right or left. And you show various movies, which actually weren't movies in those days. They were actual physical things moving around. And you ask how the choices of right versus left depend on motion in the visual stimulus. And what they arrived at was a view in which, so here what you're saying is that when you move, your spatial and temporal derivatives are proportional to each other. But a slightly gentler version of that, which emerged from the data, was that they're correlated with each other. So that if you ask, is the image intensity that I see here and now correlated with the image intensity that I see shifted over in space a little back in time, and you symmetrize it, and it would be useful if when you symmetrized it, you actually didn't make typos so that you could tell that it was symmetrized. Sorry about that. So essentially what this is saying is, okay, making them be strictly proportional to each other, that's too hard. So let me just see whether the spatial and temporal derivatives are correlated with each other. And in insects, this is really easy to think about because the pixels are obvious, right? So you can ask whether what you see in this pixel is correlated with what you see in the neighboring pixel of the compound eye with a slight time delay. And if there is, that's a sign that something's moving. Now, the problem is that this makes obvious systematic errors. In particular, if you do this and you double all the intensities, your estimate of the velocity doesn't change. It may be a bad estimate, but at least it doesn't make that systematic error. On the other hand, here, if you do this, if you double the intensities or double the contrast, you quadruple your estimate of the velocity, and that seems like a bad thing. However, that is actually what Hachenstein and Reichart saw that made them think this was what was going on, and so you think, no, we're too smart for this. So of course we're not. So what we're going to do, just for the sake of demonstration, to convince you that something like this is what's going on, is I'm going to show you two completely random movies, one of which is slightly delayed relative to, one of which is a delayed copy of the other. And then I'm going to put them on top of each other. And you realize that if what you're doing is computing a spatiotemporal correlation, you'll think you see something moving. But I assure you that there's nothing moving. And hopefully, as they start to go on top of each other, you see them move. And if the spatial displacement is smaller, it moves more slowly. And if the spatial displacement goes to 0, it goes back to looking like white noise. And if the spatial displacement is the other way, it moves the other way. And even better, if when I added things back, I flipped the sign of the contrast, so I exchanged the black and white pixels, you would actually see it go the other way. So it really is consistent with your computing a spatiotemporal correlation, even though there's nothing moving. So as many of you know, this is the core of a very successful model for thinking about motion estimation in insect vision. But it's also, if you decorate it a little bit, it's the motion energy model that people use for thinking about vision in primates. Okay, that's not to say there weren't important steps in between, but it's conceptually similar. So to go back to the beginning, if you thought that biology was finding an optimum and approaching physical limits, why on earth do you do this ridiculous calculation, which is so easily fooled? So what should you do? And what you should do is to think in probabilistic terms, that if you're trying to estimate some feature of the world based on signals in your receptors, you should compute the average value of that feature given that you've observed the receptor signals, and this will minimize your mean square error. Of course, the receptors don't respond to the features. They respond to some raw sensory data X, light intensity. And making an optimal estimate involves then being able to, right, sort of sitting inside this, there's another integral over all the features. And there's always a compromise between making systematic and random errors. And so what I'm going to do is to talk a little bit about this distribution, which is at the core of things. So how are the features that you're interested in correlated with the data that you can actually observe? So for example, sitting in this part of the equation where you talk about how the receptor signals depend on the variable in the outside world, that's where the noise in the photoreceptors or the blur due to diffraction is sitting. But then there's also this distribution, which tells you that features and so for example, the velocity with which you're moving through the world and the pattern of light intensity that you see are not unambiguously related to each other. So that relationship is still probabilistic. And that means that there are limits that have to do with the physical structure of the world that create ambiguities that you can't undo, right? And we thought about this a long time ago in the hopes of finding our way back to understanding something about the fly visual system. Sometime back is getting to be quite some time back. And if you dig into what we were doing, you find that we sort of had to guess at the form of this distribution. So what are the statistics of movies and motion as we go through the world? Who knows? So we picked something. And, you know, of course, we tried to pick something not silly, and it had interesting consequences, which I'm not going to review for you because it was a long time ago. The thing is that now there's actually a hope of directly sampling this distribution and turning these integrals that we want to do into averages over the samples. And so what we put here is the real distribution out in the world. And we don't need to make guesses anymore. And particularly here, I should point out that we weren't the only people who were worried about this problem. This is Alan's paper from 2001, where, again, I don't think I'm distorting things too much to say that part of the problem was that there's some underlying distribution that goes into our prediction of what it would mean to be optimal. So to say it again, right, the structure of the computation you should do to map receptor signals into an estimate of some feature, if you trace it all the way through, it only depends on the statistical structure of the world you live in. And so if you don't know what that is, you can say that you have this framework that's very powerful, but somehow you can't quite make a prediction without injecting some knowledge. So what Alan and Arrow did back in those days was to try to ask, how can I reverse the process and analyze human performance in perceptual tasks more deeply to see if we can pull out something about the structure of that underlying distribution. But now we can sample it. And since we're interested in fly vision, we're just in the example of this problem in flies. My colleagues, Rob de Ruiter and Charlie Adelson, when they did this, they were both at Indiana University. Charlie's now with us in Princeton. They built a camera with a hexagonal lattice of photodetectors whose spacing, optics, and spectral sensitivity match those of a fly's eye, which is pretty cool. Except you'll notice that the object is rather larger than a fly, which means that the collecting area is much larger. So this camera sees the same thing that the fly would see, but with thousands of times higher photon counts. So that means that you have essentially ground truth about what the fly should be seeing. And you instrument this camera with motion sensors so that you know how it's rotating as it moves through, as you move it through the world. And so to get at your question, what we're going to think about as motion as being in this case, self-motion, which is a reasonable approximation if you're walking on a reasonably calm day through the world and you're jiggling around at tens, if not hundreds of degrees per second. So I'm going to give you one summary piece. There are 2 generations of this effort spread over 5 years. And the idea is very simple. There's a problem of integrating information across the whole field of view. That's very complicated. So let me invite you to think just about the local problem. I look at a region that has a couple of pixels in it, and I can compute a spatial derivative and I can compute a temporal derivative. And if I thought that what the right thing to do is to take their ratio, then if I drew a contour of constant velocity, it would be a straight line in a plot of spatial derivative versus temporal derivative. And we take the logarithm of the intensity to get rid of any information about the absolute brightness of the image. And so locally, the only things that can contribute to your estimate are these derivatives. And you can ask, for every combination of spatial and temporal derivatives, what is the average angular velocity at which you're moving? This is, of course, really a 2- or 3-dimensional problem. I'm talking about what's going on in 1 dimension, just for simplicity. And what you see, so this is the old version, which you'll notice is a lot blotchier. This is the new version, which is a lot cleaner. What you see is that if the spatial gradients are large, then the contours of constant velocity really are straight lines, just as you'd expect from the ratio of derivatives. But when the spatial gradients become smaller, these contours turn around, and they start to look more like a contour of constant velocity is a constant product of spatial and temporal derivatives. And that's exactly the correlator model. And in order to get at this more clearly, what you can do is to take slices, right? So I could go to constant temporal derivative and take a slice of your best estimate as a function of spatial derivative. And what you see is this. So there's this wide range of spatial derivatives in which your estimate of the velocity is proportional to the derivative, as if you were multiplying. But if it gets to be very large, it becomes inversely proportional to the derivative, as if you were dividing. Whereas if you slice the other way, you only see the linear part and then some saturation. What's important is that most of the data is in this regime. So what we've done here is to draw a box that contains 90% of the data, or possibly 99, I don't remember. So that means that when you just go take a walk in the woods, most of the data you collect about spatial and temporal derivatives, if you try to estimate velocities from those data, they're terribly ambiguous, and the best you can do is to do the computation that Hasselstein and Reichardt proposed 70 years ago, despite all of its systematic errors. So this is just, since I can't remember, that's why I wrote it down. This is 99% of the data. So what this is telling you is that there's a chance that even these sort of dramatic systematic errors that you see in neural computation have their origins, or at least have a structure which is predictable from an optimization principle. You should just try to estimate velocity as accurately as you can. And what surprised us is not that that's possible, because there are lots of reasons to think that that's possible. Lots of people have worked on these problems. But the idea that it comes out so cleanly and compellingly from real data, right? It's not, I can imagine a statistics of the world in which this would be true. It's that the actual statistics are like this. Okay. And I think we're just scratching the surface. You can think, for example, about how this changes as a function of light intensity when things get noisier. You can think about slicing the data up to adapt to the local contrast. You can think about the problem of putting things together across the whole visual field. We think that there's a lot of what the motion-sensitive neurons in flies are doing that we should be able to extract from analyzing this. So, but that's for another day. So this was one example of the idea of optimization where I think it's because we have this new instrument and because we're thinking about it in flies and now we know how all the neurons are connected to each other in flies, it's a good time to go back and ask ourselves, can we get to the end of the problem and decide whether this view is right or not? And so far, the signs are good. But what about the more general situation? So should you believe that optimization is possible in general? And the answer depends on what you think the landscape looks like. So if I plot some measure of functional performance, for example, the reliability of your estimate of the velocity, or minus your mean square error. If as a function of the parameters inside your network of neurons, the landscape is very rough, you can believe that evolution would have a hard time finding its way here. And there are several reasons for this. One is that if you found yourself here, you'd have to get over lots of high barriers. The other thing is that if you found yourself here, you might think things are good enough. Anyhow, I think this is in fact the more widely held intuition, right? The problem of optimization is hard because, to use the language from our friends in applied math, they're high-dimensional non-convex optimization problems, right? And when I was in school, we were taught, don't do those problems, right? Unfortunately, sometimes that's actually the thing you want to know the answer to, so you have to go talk to somebody else, but never mind. So the other possibility, the extreme alternative, is that as a function of parameters, functional performance is very smooth. So if you have some way of, taking a random step one way or the other and checking either through selection in evolution or, when you're doing Monte Carlo on the computer, have things gotten better or worse, you can find your way up here without too much difficulty. We can have a more technical discussion about, will you find your way exactly to the top or you jiggle around a little bit? But clearly here, finding an optimum is not hard. And here, especially in the high-dimensional version of this, it's very hard. And so it's this high dimensionality that we decided to think about. And it took lots of people because we thought about lots of different examples. And so there's a paper on the archive, which is winding its way through the review process. And let me start. This discussion actually began with Caroline Holmes, who more or less on the first day of graduate school, she's a postdoc now, but when she first arrived as a grad student at Princeton, she asked, if I look at an insect visual system, I see nearly crystalline reproducible structure from animal to animal. If I look at our visual system, I see reproducibility of cell types. I see motifs in connectivity, but I don't see the kind of perfection that you see in an insect. And so why is that? Is it that it's not possible developmentally? Well, that's not fair, because it's not like evolution forgets about how to do things, right? So if you could do it in a fly, you could do it in a vertebrate. You just don't. So is that because they're solving a different problem? Is it because the trade-off between being able to solve some short-term problem versus being able to learn to do many problems over a long life? Anyhow, I don't need to reconstruct all the arguments for you. You get the idea. I see these two things that at first sight are solving the same problem, and yet one of them is incredibly ordered and the other one is very disordered. So how do I think about that? And in particular, is the disorder prima facie evidence against any notion of optimization? Because it seems like the ordered solution would be better. So after some struggle, we found a very simple but very concrete calculation you can do. Suppose that you look at the eye of a fly, or you look at the retina of a human. Here you see a regular array of photoreceptors. Here you see something more disordered, and to make our life easier, we're going to imagine that they're at the same density, but they're just completely random. And let me take these two receptor arrays. Imagine that each photoreceptor has some noise level, and they look out at images which have the statistics of natural images. And let me ask, how much information does this array capture versus this array? And of course, it depends on the signal-to-noise ratio, but you'll notice that over the full range of signal-to-noise ratios which are relevant in either our retina or the flies, the difference is very, very small. This came as a complete surprise to us. I don't know if it comes as a surprise to you or not. It's basically saying that, well, in some way, when you look at the fly and you see this beautiful regular array, and you look at us and you see this thing that's messy, you think, how on earth can we get away with being so messy? Wouldn't we be better if we did that? And by the time you're finished with the calculation, you realize the question is, well, what was the pressure on the fly to be so careful? Because they only gain a few percent. The answer is that in us, right, we can change the statistics of the receptors without changing the lens. Whereas in an insect, they're tied together. And that produces an effect on the optics, which is of order 1. And so it means that it's not quite fair to make this comparison. But what it illustrates is that you can be very close to optimal performance with, if you think of the parameters of this system as being the positions of all of the receptors, you can vary those parameters by an enormous amount, in fact, as much as is possible, and still only lose a tiny bit of performance. So the set of settings, of the ensemble of functional settings that come within epsilon of optimal performance is gigantic. So how do you think about that? The way you think about it is you imagine that your performance is a function of parameters. There is an optimum which maximizes your performance, and now you expand around this. You know that the first derivative is 0 because you're at the optimum, so the first interesting question is, what does the second derivative look like? What's the curvature of performance in the neighborhood of the optimum? That's described by a matrix, which is usually called the Hessian. And what matters for thinking about this are the eigenvectors and eigenvalues of this matrix. The eigenvectors have the interpretation of being the combinations of parameters that give you more or less independent variation in performance. And the eigenvalues are how sensitive you are to each of those directions. So for example, if you go back to the case of the array of photoreceptors, what you find is this. There are some combinations of variations in position, which are really produce very large changes in the amount of information that you get. But there's a sort of whole spectrum down here of modes where you can shift the photoreceptors around and you don't do very much to your performance. And in fact, these different modes, right, the problem is translationally invariant. And so these modes are basically waves that pass across the array. And so these are the long wavelength ones and these are the short wavelength ones. That's a technical detail, but the question is this structure common or not? What this is saying is that, or the guess from this, is that the reason that you can get so close to the optimum while shifting things around so much is that there are a lot of directions, in fact, in some sense, most of the directions, in which if you shift in those if you change parameters, combinations of parameters in those directions, it just doesn't do very much. And that's not a statement about biology being willing to tolerate being merely good enough. It's a statement about the structure of the problem that the organism is trying to solve. So let me give you another example. Some of you will know the beautiful work from Eve Marder and Larry Abbott and their colleagues over the years, building models of a very simple circuit. Eve would kill me if she knew that I said simple. of a small circuit in the crab's somatogastric ganglion that produces a rhythm that the crab uses for grinding its food. It's a classic problem, right? And their investigation of this system has uncovered all sorts of beautiful things, which I'm not going to talk about. What I am going to talk about is that there are models for the electrical dynamics of these neurons and synapses, which are incredibly faithful to the real data. And they consist of giving each cell several different kinds of ion channels and then hooking them together with synapses. And the dynamics depend, among other things, on how many of each kind of channel is present in each cell. And since each cell makes 5, 6 different kinds of channels, and there are 3 cells in this little sort of pared down network, there's 20 parameters. There are more, of course, because the animal chose those particular channels out of a genome in which there's 100 different kinds of channels it could have chosen from, right? So we're not even talking about that. We're just talking about you're committed to making these channels. How many copies of each one do you put into the membrane? And you can say, well, how do I measure performance? You could say, well, I could measure the period of the oscillation. That seems reasonable. So let me choose as a measure of performance the mean square difference in period from my target, normalized so that it's a fraction. Or you notice that an important part of the rhythmic structure is that there's a duty cycle, right? This neuron is on for some fraction of the time and then off before it turns on again. So you can think about the duty cycle, and you can do that for each of the different neurons. What's interesting is that it doesn't matter which of these 4 things you pick, the answer is the same. The spectrum of eigenvalues of the Hessian matrix dies exponentially and then actually crashes at the end. And this means that it spans, well, if you really want to include the crash, it spans 10 orders of magnitude. And if you're a little more conservative, you would say, well, sorry, this is 10 orders of magnitude here. Right? So what this means is that there are combinations of the channel densities, combinations of the protein copy numbers that you put in the membrane, which produce an effect. Let's say this is of order 1. There's a square. So this is 10 orders of magnitude smaller. It means that a displacement here, the natural scale is 10 to the minus 5 smaller. Right? So if you think that you need to control these variables with a precision of 1, you only need to control these with a precision 10 to the 5th, which sort of means you don't need to control them. Right? This kind of structure has appeared in many other places. And let me, in particular, Jim Sesson and his colleagues at Cornell pointed out that this structure comes out when you try to fit models to data. So if you have some reasonably complicated model, and by the way, the prototypical example of a complicated model is a sum of exponentials. So if I show you something that's decaying, and I say fit it with a sum of exponentials, each of which has an amplitude and a decay rate, which you can imagine maybe you did in some introductory chemistry course, it turns out that that problem is not very well posed. And it actually has this structure. If you look at the sensitivity to the different combinations of parameters, as the model gets more and more complicated, you end up with this thing where the distribution of sensitivity to parameters, you have to plot it on a log scale. And it's nearly uniform on that log scale. In their hands, they were thinking about fitting a model to data. And so if you had parameters to which the model was not very sensitive, the argument would be, oh, that means my model is too complicated. I should somehow rewrite the model and take that parameter out, or just push it to its edge of allowed values, and that would reduce the complexity of the model. The point of view we're taking here is that the thing you're measuring is not quality of fit to the data. The thing you're measuring is a functional performance measure that the animal actually cares about. And the parameters aren't things that you introduced in building a model. They're real things that the biology is doing, like putting copies of a protein in membrane, or having a certain binding energy for some transcription factor to a particular site in the genome, right? And so it's not that if those parameters are poorly determined, that you should remove them, but rather it gives space for variability in parameters across organisms without loss in performance. Let me give you another example, which is sort of fun. What we did here was to build a very simple recurrent linear network and read out from that network. So this is a completely linear problem, sort of boring. And what we ask for is that the output is necessarily a filtered function of the input, and we ask that the filter be something in particular that we picked. And then we just go through the same machinery. You could view the parameters as being the weights in reading things out, and then you see a structure which again has this variation in eigenvalues that spans 10 orders of magnitude, despite the fact there's only 100 neurons. Or you could view the readout weights as being fixed and ask what happens if you change the internal connectivity of the network. I will point out that if you trace the mathematics through, the only thing about the internal dynamics of the network that matters are the eigenvalues of the matrix and not the eigenvectors. So although if you have 100 neurons, there's 10,000 connections, it turns out that only 100 numbers are actually constrained by trying to match a particular input-output relationship, because you can always undo the effects of changing the eigenvectors by changing the readout weights. Okay, so we knew that, and so you would say, oh, you might have thought it was a 10,000 dimensional problem and it's only a 100-dimensional problem. I've made a discovery. No, you haven't. It's just linear algebra. The surprising thing is that of the 100 dimensions that are left, the sensitivity varies exponentially. That wasn't obvious at all. These linear problems are a little boring. So it turns out that there are echoes of this in nonlinear problems. One now some years ago from Kanika Rajan when she was still in Princeton, and more recently from David Clark, who just finished his PhD at Columbia. Let me say this one in words that are as close as possible to what I was just talking about. If I want to encode a periodic variable like the direction in which my head is pointing in a population of neurons, I want the activity to have a sort of bump which represents the direction, and it can move smoothly as I turn. And so you ask yourself, how do you do that? And the natural answer is you connect neurons to each other so that the ones that represent nearby directions excite each other, the ones that represent much further separated directions inhibit each other. There's a stable solution, which is a bump, but the bump can be anywhere along the circle, and that's how you do it. That's a very ordered structure. And what these guys found is that you can have incredibly disordered structures that also work. And one way of saying that, one way of understanding that in relation to what I just said, is that what it means to represent a variable that lives on a circle is that there are two dimensions that have a kind of degeneracy, which if you think about it, corresponds to having degenerate eigenvalues in the synaptic connection matrix. And as long as that's true, you can pull out of the dynamics a representation of a circular variable. But saying that two eigenvalues are degenerate, it's a strong constraint, but it's only one constraint, and there's n squared connections. So in fact, there's a huge volume of networks that can achieve something that you would normally think of as being achieved by a very ordered structure. Same idea. Let me do one more, and then I'll wrap up. It turns out that there were hints of this in the artificial neural network literature, although they didn't make the connection to what Sethna and company had done about what they called sloppy models. And so we went back to try to make this a little bit more precise. So here, There's, by modern standards, a relatively small neural network that's been trained on the CFAR image classification task. It has some relatively conventional convolutional structure. You can train it and it learns. And then if you do, it has 100,000 parameters. And so if you do the analysis that we just described, and instead of plotting the eigenvalues in rank order, let's look at their probability distribution. And their probability distribution is a power law, which extends to the smallest eigenvalues that you can reliably measure. By the way, I'm hiding the technical developments that made it possible to do this calculation. You do not, you will be shocked to know, diagonalize a 100,000 by 100,000 matrix. Okay, there's a trick, okay, which we could talk about. Importantly, this structure emerges during learning, which, if you could read the labels, you'd know. Sorry about that. And perhaps more interestingly, if you randomize the categories to which each image is classified, the network can still learn to do this, but it doesn't have this full scale invariant structure of the eigenvalue spectrum. It can still learn to classify. So it can take hundreds of pictures of airplanes and call some of them cats systematically because that's what I told you to do. But then, and actually you can do surprisingly well, it takes you longer to learn, which may be interesting, right? If I give you some completely scrambled version of the problem, it's hard to learn, but eventually you do. But then you don't have this structure of widespread ranging sensitivity. And this is not just a consequence of overparameterization. Some combination of parameters, you have truly zero sensitivity because you don't have as many data points as you have parameters. But this is about the ones that are left. Okay, so let me try to synthesize and then stop. So let's imagine that I'm an organism and I have something I'm trying to do and I have a measure of my functional behavior. And it depends on parameters. But I don't know, neither I nor my evolutionary history really know how to set those parameters exactly. So let's imagine that they're drawn from a probability distribution. A plausible guess for that probability distribution is that it's the exponential of your performance, both because maybe you are thinking like a physicist and you say, well, it's like being in equilibrium and the performance measure acts like the negative of the energy. Or you could say that I want to be sure that the average performance in the ensemble is a certain value, but otherwise I want to leave the parameters as much room to vary as possible. And that turns out to be the same thing. So let's take this as a model, and then let me hide some algebra from you in which we imagine that in the neighborhood of the optimum, the eigenvalue spectrum has this form where each eigenvalue is sort of exponentially smaller than the last. And then let me grind through and ask, what is the average performance? Well, it depends on beta, and it depends on the spectrum, and it depends on this coefficient alpha, right? It depends on everything. But you can rewrite it in an interesting way, which is that it involves an exponential of the entropy per parameter. So essentially, I'm taking the volume of the parameter space that is available to me, given that I have some average level of performance. I'm taking the logarithm of the volume, and I'm dividing by the number of parameters. So one way to think about it is that if each parameter could vary by a factor of 2, then this number, S over K, would be log 2. Okay? And of course, it's not a single parameter varying, it's combinations, so it's a little more complicated than that. But that's the idea. In particular, if this number is finite, it means that if you pick a random direction in parameter space, you're going to see substantial variations in the value of that parameter. Or sorry, along that direction. But importantly, if you have this exponentially decaying spectrum, you get another term in your performance, which is exponentially decaying. And Here, things depend actually on the total number of parameters. And so if you keep everything the same, but let the number of parameters get larger, eventually this wins and you're at the optimum, despite the fact that you had finite entropy per parameter. So what's the summary? The main summary is, as I've said already, that in a high-dimensional parameter space, you can get arbitrarily close to optimal performance despite the fact that the parameters are highly variable. This is not about being good enough. It's about the structure of the problem that you're trying to solve. And that structure allows you to have this coexistence of optimality and parameter variability. It's not just about overparameterization, because this is about the parameters that are left after you've accounted for the overparameterization. And of course, we should add that we've only said things about the local structure of the landscape. There's still lots of open questions about the global structure, which I'm just not going to say anything about. And what we hope is that these results are on the way to removing one of the major conceptual obstacles to thinking about optimization as a more general principle. That it just isn't as hard as we thought it was to be close to optimal in problems that actually matter in the life of the organism. You can make up an artificial problem for which it would be very hard, but in these examples, and there are more, which have something to do with the problems that organisms really have to solve, it seems like those problems have this structure which allow for this coexistence of optimality and variability. So thank you for your attention. Yeah.
And that one step forward.
Mm-hmm.
So you say that this depends on a variety of things, including natural image statistics. I guess computers like it.
Yes.
And,
Right. So this is a thing that we compute from the statistics that we see. Right. Right.
how does it analyze
Yeah.
changes?
Or we can sample them in the real world. Right. Well, so we can certainly do this problem where we make up different statistics. And arguably that's what we did 30 years ago. And what I can tell you is, so 30 years ago, we had a model for image statistics, which was surely wrong in detail. Actually, probably wrong even before you get to details, because we just made it up. Sorry?
You'd be very lucky.
Right. And that model had the property that there was some limiting case where things looked correlator-like, and some limiting case in which things looked ratio gradient-like. And so I can remember our conversation at the time. We said, this is fantastic, right? Sometimes you do one thing, sometimes you do the other. In fact, if you look closely at the responses of neurons in the motion-sensitive neurons in the fly visual system, at low contrast, they're unambiguously doing something correlator-like. At high contrast, the firing rates of neurons are still being modulated by the velocity. So things have saturated as a function of contrast, but they're still giving you a veridical encoding of velocity. So maybe that's a crossover. So we thought, this is all wonderful. There's only one problem. The location of the crossover point, even in the theory that we wrote down, is a parameter that we're free to choose because we don't know it. So the important result here at this stage is maybe not that it's correlator-like in here and ratio of gradient-like out there, for which there are many general arguments. I'm not sure that we understood that completely 30 years ago, but okay. The thing that's interesting is that almost all of your experience is inside the box where things look correlator-like. That's the thing that you can't get by just sitting there and thinking about it. Now, one way to change, right, that the bulk of your experience is in a regime where you do the weird thing. You're forced to do the weird, or you would like to do the weird thing that seems so paradoxical. I'm not sure, I mean, we can change the image statistics by going for a walk in a different place, but maybe deeper is we can add back, remember that the camera is collecting 1,000 times as many photons as the fly is. So we can add back the noise that comes from real light intensities. And what that's going to do is to take this region, which is correlator-like, and expand it out. It also, I haven't talked about the question of whether you should do averaging over time when you compute these gradients. And of course you should, but how much you should average depends on how noisy things are. And those are all things that we can get at, you can get at in the responses of the neurons and have people have for years and years and years. We should have concrete predictions for all that by going through the parallel analysis and those different. We're hoping to sort of unfold these various, if you want, sort of, I don't know, context-dependent or adaptation kinds of changes in the nature of the computation that the insect does. Yes. Mm-hmm.
I'm not sure if I'm understanding correctly or I'm just mismatching the concept.
No, well, so I think if you go back, so let's look at a example here. You could say, so I've made a model of this neural circuit. And there's some combinations of parameters which don't matter at all. So shouldn't there be a representation of the model in which those parameters aren't there? So somehow, so this is not about sparse encoding of the world outside, but sparse encoding of your description of the circuit, right? And so I think that was the point of view that Sethna and company had, which was that when you see this, it sort of invites you to simplify the model. Say, look, if I only care about predictions with some accuracy, then I can take a whole bunch of parameters and set them to whatever value I want. And in particular, I can set them to values that simplify all the equations. And so then by hand, I've found a simpler model, which I know will perform almost as well. So that was the direction they were going in. I think our point of view is a little different. I would have to think. So for instance, here in this artificial network solving a vision problem, I don't actually know this, but I think the conventional wisdom would be that if I stuck my hand in one of these layers and I looked at the distribution of activity of the individual units, it would be sparse in the sense that the distribution would have a very sharp peak near zero and a tail. I don't understand the relationship. It feels like that's actually different than this, but they can't be totally independent of each other because this is about derivatives with respect to the strength of connections, and if you change the strength of the connections, you change the activity. So it's a good question. I don't know. Yeah.
Yes, you ended the talk by saying essentially, you paraphrase if I get it wrong, but that if biology has enough mechanisms that it's supposed that biology's got more than one way to skin a cat or achieve your optimal functionality, right?
Actually, you might want to be a little careful about whether it's many mechanisms or one mechanism that is rich enough.
Well,
and I don't know which one you want.
Mechanism, for example, I'm equating mechanism with ion channels.
Right, and so there,
So where I was
yeah.
going with the question is, so you presented this as a way of understanding how neuroptimal performance variability.
Yeah.
You also presented a plot showing a non-convex fitness landscape versus
Right.
quite convex fitness landscape. And I'm wondering if you're thinking of the issue that you concluded your talk with as a reason why it should be more easy to pop out of a local optimum or not. Or
Okay, so, or I possibly misled you. So at the end of the day, so the point of drawing a non-convex landscape was to sort of tap into a widely held intuition for why optimization can't work. In fact, nothing that I've told you illuminates the structure of the global landscape. So in the neighborhood of the optimum, it's always convex, right? Non-convexity is a global statement. Now, our friends who think about artificial neural networks, they've tried to think harder about the global structure of the landscape. And so they had results which presage the ones that I just showed you, that in the neighborhood of the optimum, there are lots of directions that are very flat, which is what corresponds to having these very small eigenvalues. But they also, right, you train a network and you can, exactly what parameter settings you get depend on which random examples you drew out of your data set and even which order you presented them in, to give a nod to the evolutionary biologists' worry about historical constraints. So you can do things like train two networks, and at the beginning, you make the same random choices for the data. And then at some moment, you let them diverge. And what you find is they end up in different places, both essentially for a large class of problems, the sort of image recognition classification problems I'm talking about, essentially perfect performance in both cases. Not only is it true that in the neighborhood of each optimum, there are lots of flat directions, but in some cases, if you just draw a line from one set of parameters to the other and go to the middle, that model is pretty damn good too. So that's telling you that even if you have local optima, which are distinct, there are very smooth paths that connect them. I don't think we have a full characterization of these things. But this and many other results from that field are telling you that I think people imagined that the problem was getting stuck in local minima. And I think we should be grateful that the people who first trained these networks didn't know that they were not supposed to do non-convex high-dimensional optimization problems, or we wouldn't have gotten anywhere. And the answer must be, it is true that somehow some version of the generic non-convex high-dimensional problem is not solvable. But there's something about the structure of these that makes the landscape much more navigable. So I've focused on one thing which is related to that, but there's more to say.
Why you can get many solutions near a given optimum.
Well, near I can tell you about.
Right.
It's separated by a barrier. I don't have anything to say. In thinking about the genetic networks that are relevant for patterning in embryos, we've actually done something related to all this. And if you look for networks that convey as much information as possible with a limited number of molecules, which limits the noise level, you find local optima and that are approximately degenerate. Or maybe they differ, for example, in you gave a budget for the maximum number of molecules and they differ in the mean number of molecules. We don't really know what you should have constrained. But there are hints that, there's a clear result that there are local optima that look a lot like the real network that we were studying, which is surprising because we didn't put in anything. But if you look at other local optima, they look a little bit like the networks that you see in closely related species. And if I could tell you that that was true, then I would stop, right? If I could tell you that not only are there lots of local optima, but they're actually represented out there on the tree of life. Okay, that would be. Yes, we should not end on such an optimistic note. Let me take one more question. Really? Mm-hmm. Um, so the so yeah, I could answer yes, you're right. We need to think about this, and that's true. I would emphasize that in many of the examples I show you, what you mean by performance is already an average over many instances of the problem. So by changing the distribution of those instances, you're not solving a different problem, but you're solving a problem in a sufficiently different context that it almost feels like a different problem. Let me also point out something which is what one of our reviewers is worried about. So we said, well, how do I measure performance? I could measure the period of the oscillation. I could measure the duty cycles of the individual neurons. And it sort of doesn't matter. So this is not as good as what you want, but it's at least, given what we were calculating, this is the direction I think that you're asking about. And our reviewer's comment was, very nice, but what if you needed to do all of them at once? Then surely you wouldn't be able to tolerate as large a parameter change. And I don't know the answer to that question yet. So there clearly is, I mean, in some way, I think the thrust of your question was, I have to solve many different problems with the same machinery. Which way does that cut, right? Does that mean that I'm allowed greater variation because some parts of the space will be good at some things and some parts will be good at others? Or does it constrain things because I have to do two different things and that's two different constraints? We don't know. Okay. Yeah, I don't know is always a good place to end. Thank you.